Hi everyone! In this video, we gonna learn about immunological tolerance. So, let's get started. Basically, our immune system can detect or recognize many kinds of antigen and that can thus stimulate the immune response. So, what is the immunological tolerance? Immunological tolerance can be described as the specific immunological non-reactivity to an antigen due to the exposure of same antigen. For example, the immune system is generally tolerant of self-antigen, so it does not easily attack the body on cell, tissue, and organs. However, tolerance can be induced by certain type of antigen that called tolerogens. Same antigen can be both an immunogen or tolerogens. It depends on how and where it is presented to the immune system. Next, tolerance can be classified into central tolerance or peripheral tolerance. It depends on where the state it is originally induced. For example, central in thymus and bone marrow. For peripheral, in the other cell or lymph nodes.
Central tolerance occurs in early maturation stage. It occurs when there is a lymphocyte cell that recognizes self cell and try to kill it. So imagine central tolerance as Mr. Policeman that tried to get rid of this culprit and allow the entry of the self cell into the next maturation stage. Central tolerance is also called as negative selection, and it is carried out using two mechanisms. The first one is clonal deletion through cell apoptosis, and the second one is receptor editing. Clonal deletion occurs when the immature lymphocyte expresses receptor toward the self cell. Now imagine the knife as the receptor that causes the lymphocyte to have high affinity and ability to destroy the innocent self cell. This kind of lymphocyte is so dangerous that it will be deleted through cell apoptosis, and the self cell will survive. And this clonal deletion occurs in primary lymph organ, which are thymus for T cell and bone marrow for B cell. The second mechanism is receptor editing. Receptor editing are the mechanism used only by immature B cell in order to get away from cell apoptosis program. It involves the process of changing the antigen receptor of the cell-reactive immature B cell so that it will not recognize and harm self cell anymore. How does receptor editing work? So what actually happens in receptor editing is that the V and J gene in the light chain of the antigen receptor is modified in different arrangement, therefore causing the receptor to have different specificity and may not recognize self-antigen on the self cell anymore.
Next, we are going to learn about peripheral tolerance. It happens because unrecognized self-reactive lymphocyte and weak self-reactive lymphocyte have entered the circulation during negative selection. Two main events that happens during peripheral tolerance are suppression of immune system by regulatory T cell and induction of energy T cell by CTLA4 molecule. Regulatory T cell or Treg cell will help to recognize self-antigen and downregulate or suppress the immune response. By doing that, Treg can protect self-antigen from being attacked. For T cell to be activated, they need to bind to the antigen presented by MHC and undergo co-stimulatory engagement by having interaction between CD28 receptor on T cell and CD80 or 86 receptor on antigen presenting cell. In peripheral tolerance, CTLA4 molecule inhibits the interaction between the receptor to avoid formation of co-stimulatory signal. Therefore, T cell cannot be activated, thus producing only T cell energy. T cell energy is an inactive form of T cell, so it cannot attack self cell antigen.